Hello 515 students. This short video will show RNA extraction from yeast. In the last video, we cultured yeast in growth media that had either glucose or galactose as a carbon source. We will extract RNA so that we can perform transcriptomic analysis and compare gene expression in yeast grown in either condition. We will show you the steps to isolate yeast RNA using the QIAGEN RNeasy mini kit. This is similar to the procedure you previously used to isolate RNA from E. coli, but there are some differences. First, we resuspend the cell pellet in RNA lysis buffer. This is done for both cells grown in glucose and cells grown in galactose. Next, we need to break open the cells. Yeast have cell walls that differ in composition from those of E. coli, so we can't use the same enzymes to break them down. Instead, we will use glass beads to break open the cells with a bead mill. We add the cells suspended in RNA lysis buffer into tubes containing glass beads. We do this for the cells grown in glucose and those grown in galactose. Then we place the tubes in the bead mill, running it for 1 minute, then cooling the tubes on ice for 5 minutes. This is repeated 5 times. After breaking open the cells, we remove the cell lysate from the glass beads and place it in a new tube. And to make sure that we get rid of all the beads, we centrifuge the tubes and pipette the lysate into another new tube, leaving any beads at the bottom of the previous tube. We do this for both samples. We then add 1 volume of 70% ethanol to each sample. Then mix well by pipetting up and down. This mixture will bind to the RNeasy spin column. After applying each sample to a spin column, centrifuge them. The flow-through that goes to the bottom of the collection tube is discarded. Then we add a volume of the first wash buffer to the spin columns on which the RNA is bound. Centrifuge. Discard the flow-through again. We add another volume of the first wash buffer to each column, then spin again. Discard the flow-through. Add a volume of the second wash buffer to the columns, and centrifuge as before. Discard the flow-through again. Finally, each spin column is placed in a clean 1.5 milliliter Eppendorf tube, and RNA's free water is used to elute the RNA. The eluted RNA is then collected in the bottom of the tube by centrifugation. The purified RNA samples can then be stored at -80 degrees Celsius for later transcriptomic analysis by RNA-seq.